They were really hard to read with the picture that I had, so I just removed the picture. By the time we hit the rest of these slides, I'll find a picture that will help us to read these a lot better. All right, first 4 questions. What are the 3 steps of transcription? What are the 3 steps of transcript modification? What is unique about transcription in eukaryotes? And how does this transcript modification add complexity to the design of a eukaryotic organism? All right. Before we start addressing these 3 questions, I want to draw you what we call the central dogma of molecular biology. Okay, the central dogma of molecular biology basically says this. You take DNA, you transcribe it. I don't know what that H is there for. You transcribe it. Oh man, I need an S. You transcribe it into RNA, which is then translated into protein. This, once we understood that DNA was the molecule necessary for heredity, we knew what its structure was, we discovered RNA, and we basically was like, this is what DNA exists for. It exists so it can be transcribed into RNA that can then be translated into protein. Okay? This is a central dogma of molecular biology. What we find now is it's way, way more complicated than that. It's way more complicated than that. But we're going to start at this first process, when we are transcribing DNA into RNA. Okay? We'll start there. What are the 3 steps of transcription? So again, transcription is writing RNA from DNA. True or false? RNA is a nucleic acid. True. True or false? RNA is structurally similar to DNA. Also true. So true or false, writing RNA from DNA is going to have some similarities to copying DNA. Also true. In fact, copying DNA, you sort of do this in that you read the DNA template and you write an RNA primer, right? That then has to be replaced. And so we basically have 3 parts of RNA polymerization or of transcription, whatever you want to call it. The first is initiation. And here we have what are called transcription factors that bind to what we call a promoter region. Upstream from the part of the DNA that's going to be transcribed, written into RNA, there's a promoter region that these machines called transcription factors bind to. In eukaryotes, we call this the TATA box, because the sequence in this promoter region is what? What do you think it is? TA, and then AAA. And so since this is a sequence, we call it the TATA box. In prokaryotes, they have a different promoter, but they still have promoters upstream of the DNA that's going to be written. Then the DNA unwinds, similar to when DNA is polymerized. Now the difference here is helicase is not involved. The machinery that writes the DNA also unwinds it. RNA polymerase unwinds the DNA and reads it and writes it. Very complex molecular machine. Then RNA polymerase, look at this, which is self-priming. And what does that mean? It does not need a primer. It can add that first nucleotide. It doesn't need a free carbon 3 to attach a nucleotide like DNA polymerase does. It's self-priming. It gets in, it attaches to the template strand. All of this happens during initiation. Okay? Transcription factors bind the promoter. RNA polymerase unwinds the DNA, attaches to the template strand, and now initiation is over. And then step 2 starts. Step 2 is called elongation. And in elongation, DNA polymerase, or RNA polymerase 2 if we are writing mRNA, but we'll get to that in a minute, RNA polymerase II comes, it's already attached to the template strand. It's already unwound the 2 strands of DNA, and it starts reading the template strand and then writing an RNA copy to that template strand. And so in elongation, it just keeps moving down, reading that DNA template and writing an RNA copy of it. And that's what happens during elongation. Now, it's really complicated how it's working. You could probably imagine that because we talked about DNA polymerization, and you're like, there's a lot of machines involved, and it's complicated. But here, notice we are only writing 1 strand, meaning you don't have to worry about what? Discontinuous and continuous, because you're only writing 1 strand, and you're only writing the strand in the direction that you can read it and write it. So we don't have to worry about Okazaki fragments and pulling it through backwards. It's just it's reading it and it's writing an RNA complement. So if there's a adenine sitting right there on the DNA, it attaches the complement to that. Which if it were DNA, that complement would be thymine, but there is no thymine in RNA. Instead it's uracil, okay, which is the RNA version of thymine. All right, the last part is called termination. And in termination, the RNA transcript and RNA polymerase II are released from our DNA template. Now, the particular mechanism of how that happens varies from organism to organism, but ultimately the whole thing becomes destabilized. It reaches a certain point as it's writing that it writes something in the RNA transcript that destabilizes the whole complex, and it all falls apart. Releasing the RNA, releasing RNA polymerase to go and write new RNAs, maybe at that same section or some other section in the genome. Cool? Yeah, Allison.
So does the DNA be attached to it?
It does. Yep. Yep. Once that RNA strand is gone, the DNA reanneals. You call it reannealing. It comes back together. It unmelts. Because when DNA is separated, we call that melting. Yeah, Micah.
So the top box is on DNA?
Yes, the Tata box is on DNA. And here, I'll show you that here. So here's the portion of the DNA that's going to be transcribed, that RNA polymerase is going to read and write an RNA version of. Upstream from this, we have the promoter. And in the midst of that promoter, we have the Tata box, T-A-T-A. Okay? That's why we call it the Tata box. The transcription factors bind there during initiation. They recruit RNA polymerase that unwinds the DNA and attaches to the template strand. Then elongation takes over. Oh, here are those transcription factors here in our RNA polymerase II. And so now initiation is done. And then we go into elongation, where RNA polymerase is reading the DNA template and adding RNA nucleotides to make an RNA version of it. And it just keeps going down and just keeps transcribing. And look at this. Parts of the DNA have already reannealed, recombined, back behind where the active polymerization is happening. And it just keeps moving down all the way until we get to termination. And then there's something in the strand of DNA that when it writes an RNA version of it, destabilizes the whole process. RNA polymerase falls off. The RNA transcript that you just wrote, the RNA version of the DNA falls off, and now it can be used to do something else. According to the central dogma of molecular biology, what will it be used to do? Translate into protein. Okay? It's a lot more complicated than that. Less than 2% of our DNA adheres to the central dogma of molecular biology, that it's transcribed and then translated into protein. About another 85 to 95% of it gets transcribed into RNA that is never translated into protein, but instead has a regulatory role. Chris?
So for every gene that needs to be transcribed, there's always a Tata box?
Yes, in eukaryotic genes.
Okay.
Yep, in eukaryotic genes. Yep, you got it. And then that way the same transcription factor can recognize every gene and recruit RNA polymerase.
Okay.
Yeah, Allison.
So since the DNA strand is complementary, does it matter which side?
It absolutely does matter which side RNA polymerase reads and writes. Yeah, because if it needs a copy of this strand, but an RNA version of it, it actually has to read the complementary strand to write that strand. Yep, absolutely. It does matter which one it reads. Yep. Does that make sense? If it needs an RNA version of this strand here, the only way to do that is to read the complementary strand so that you can attach the complementary RNA nucleotides.
That's because the machine doesn't go with the other version.
Well, and it's just that's how this works. That's how it knows which base to put in, is by reading the complementary strand, right? Because otherwise it'd have to work under some completely different mechanism from the way these machines work, DNA polymerase, RNA polymerase included. Okay? So again, what are the 3 steps of transcription? Initiation, all the machinery shows up. Elongation, the machinery does its work, makes an RNA version of the DNA sequence. And termination, the machinery all falls apart. Okay? Falls off and releases the RNA version of the DNA. All right. Now, what we have found is that even stretches of the DNA that conform to the central dogma, something happens in between here and here that's not on this diagram. Okay, again, this is the central dogma. This is how molecular biology works. DNA exists to be transcribed so it can be translated into protein, right? Or using our analogy of the factory, white-collar workers exist to tell other white-collar workers how to make machines, okay, that can actually do the work, right? But even those parts, which is less than 2% of our DNA, actually conforms to the central dogma. Even the parts that do, something happens in between here and here. And that's this RNA gets modified. We're going to talk about that right now. 3 steps of transcript modification. So after transcription, we have what we call a pre-mRNA molecule. So this RNA in the central dogma is a specific type of RNA, and specifically it is mRNA. And so after transcription, we have a pre-mRNA molecule. That it has to be matured. It has to be modified into what we call mature mRNA. And here are there are 3 steps to this maturation process. 1, we attach a guanine nucleotide backwards at the 5' end of this. Okay, so the first step is we attach a guanine nucleotide backwards at the 5' end. And which end is going to be the 5' end, the beginning or the end? We write 5' to 3', right? So the end of your transcript is going to be what? The 3' end. Yes? We write 5' to 3', whether you're DNA polymerase, RNA polymerase, doesn't matter. That's how we write. We write 5' to 3'. So the 5' end is at the very beginning of this RNA transcript. And you attach a guanine nucleotide backwards. And you're like, that's really weird. What is a backwards guanine nucleotide going to do? It's an excellent question. We'll talk about that next week. All right. We call that the 5' untranslated region. That's what UTR stands for, 5' untranslated region. So at the very beginning of our mRNA, but a part that's not going to end up getting translated. Then at the other end, which we call the 3' untranslated region, why do we call it the 3' end? What does it end in? A free carbon 3, right? So at the 3' end, at our 3' untranslated region, we remove some of that and then we replace it with a big long tail of A's. A big long tail of A's. And we call this a poly A tail, and so the enzyme that adds those A's is called poly-A polymerase. And it adds 50 to 250 adenines at the end of the mRNA. And so this serves to protect it from enzymes. The RNA, so in here, this RNA, in order to get translated into protein, has to leave the nucleus. Okay, again, our analogy of a factory is a really nice one. Your main white-collar workers, they don't go into the factory, right? They stay in the office. You've got some white-collar workers that go out into the factory itself. In order for this RNA to be translated, it has to go out in the nucleus. Well, you know what there are in the nucleus? Or, sorry, it has to go out into the cytoplasm. You know what there are out there? Enzymes that chew up nucleic acids. You're like, why is that? Because you consume materials that have nucleic acids and we use it as fuel sources. So adding this big long tail of adenines gives those enzymes something to chew on while this is being translated. It's like when you're training those German shepherds to be police dogs, right? And so you put on that protective suit, you give the dog something to chew on, right? While you're running through the field training the dog. Have you ever watched one of those? Somebody get into that suit to train. Man, my dad worked in law enforcement my entire life. Well, not my entire life because he's retired now. But from the time before I was born up until I think I was 25, my dad worked in law enforcement. And his favorite part of his job was when they called the K-9 unit in. Because it's just everything changes at that point, right? You send a dog in where humans can't get to, and then all of a sudden the situation's diffused, right? Dogs take care of it pretty quickly. Sorry. I don't know how you feel about police dogs, but I like them. All right. On top of this, so that's step 1. We attach a guanine nucleotide backwards. Step 2, remove part of the 3' untranslated region, add a big long poly-A tail. And then the third part is we have to cut out sections within the sequence. Okay? So we've got to take sections that are inside. They're not in the 5' untranslated region. They're not in the 3' untranslated region. They're sprinkled throughout the part that is actually going to get translated, but they have to be cut out. Like, this is really weird. Why would you design something that before you translate it, you have to remove some of the words? Like, that doesn't even make any sense. Keep that stored up somewhere. Okay. So here's the process of modifying or maturing pre-mRNA. So we've got this 5' untranslated region. We're going to attach our guanine cap. We're going to take our guanine nucleotide, attach it backwards here. We're going to cut this off, and we're going to add a big, long poly-A tail that you can see here. Attaches a whole string of 50 to 250 adenine nucleotides. And then we need to cut out these introns, these parts that are not going to get translated. Again, it's like you have a message, and some of the words need to be removed before you translate it. And so here's right here is the process that cuts out these introns. We've got these proteins here called small nuclear RNA proteins, and they create what's called the spliceosome that cuts out these introns. And you see they've wrapped that up, they've looped this, they'll cut it here and just keep the parts that are actually going to be translated as part of this sequence. So we move through here, this part got cut off, gets degraded down into individual RNA nucleotides that can be used in the next round of transcription. And then these can, come off now and be recycled and be used in the next round of intron removal. Micah.
How do you go from the mixed introns to the one bar of introns?
Oh, it just zoomed in on one.
Oh, I see.
So what it did here is, here we've got multiple introns, and then here it's just showing you, okay, in an example where you only have one intron, this is how it would be cut out. But in this example here where they started with, which they should have continued with, both these introns here would require a spliceosome complex to cut them out. Yeah, Chris.
Is it possible for those spliceosome proteins or whatever to cut part of the
exon?
Yeah.
Oh, sure. Make a mistake?
Yeah.
Yeah, it doesn't happen very often, as far as we can tell, but yes. Yep. All right, so we're going to take a lecture break. We are currently, I think, 27 minutes into class. It's a good time for a break, okay? And so what I want you to do is this. I want you to give me a reason why you would have sections within your transcript that need to be removed before it's translated. What is the functional value of having introns that need to be cut out? Why have them there in the first place? Right? If they're just going to be cut out in between here and getting here, right? If they're just going to be cut out, why are they there in the first place? Okay, take a couple of minutes, talk with those around you, and I want you to give me the functional role of those introns. 2 minutes, starting now.
Oh, ready? Did you guys talk about the
So it gets sent out.
Is it for the process of like
That's right.
making sure what
Multiple RNA. So,
So I guess it's
so it's like
we attach something like this, right? In the first place. Remember, we attach something.
You have about 30 seconds.
oh yeah.
Yeah, so when their job is that you're done in them, so that's why they are cut out. It's kind of
All right. Do you have a functional role for introns? Why would you transcribe something that is never going to be translated anyways? Why would it even exist in the DNA if it's never going to be translated into protein? Yeah, Peyton.
Have a proper distance for the 5' 3', that way it keeps matching up.
Okay. Yeah, that's a good idea, right? I mean, that's a nice thought that there has to be a certain length, a certain total length to your pre-mRNA in order to give the enzymes enough room to work. Now, what's interesting is there's a huge variation in the length of these mRNAs. The smallest protein, I think, is about 500 total bases in the mRNA, and then there are some of them that are tens of thousands of bases in length. Levi.
My idea was that it might have something to do with the process of getting where it needs to go.
Okay.
Something to do with, because it's a complicated process of, I don't know.
Right, so it has to be shuttled outside of the nucleus, right? I mean, it can't. It's written in the nucleus and it needs to leave there, right? And so that might be just something required, that maybe one of those signals that you're cutting out is recognized by a machine that moves it towards. That's a really, really nice thought. That's a really nice thought. Yeah, there are machines that recognize another region of it, but it is a really nice thought. Any other functional roles that this could play? Okay. I'll give you the answer in a minute. Not right now. Okay, our next question. What is unique about transcription in eukaryotes? First of all, the TATA box, or the TATA box, the promoter sequence that the transcription factors recognize, that is unique to eukaryotes. Prokaryotes have a promoter sequence, but it is not the TATA box. RNA polymerase II needs the transcription factors in order to bind in eukaryotic DNA. It's not the case in prokaryotic DNA. They still have promoters, but RNA polymerase can bind wherever it wants and can start transcribing the bacterial chromosome. You're like, that kind of makes sense. Their chromosomes are just smaller. Just smaller. Eukaryotic DNA doesn't have a sequence in it that tells the RNA polymerase it's time to stop transcribing. And instead, there's something else that happens to destabilize the whole process. And then for our what we call non-coding RNAs, so those that do not conform to this, these are coding RNAs. If DNA is transcribed into RNA and then translated into protein, we call these coding sequences. So for those that get to here and make a different type of RNA, not mRNA, but that is never translated into protein, these have different promoter sequences. And they're specific to the type of RNA polymerase that writes them. So RNA polymerase 3 writes all of our different types of tRNA. There are 49 of them, at least in humans. Probably in all eukaryotes. RNA polymerase 3 writes the 49 different tRNAs and one of the 4 ribosomal RNAs. And this is RNA polymerase 3. RNA polymerase 2, we already talked about. What type of RNA does it write? mRNA. Specifically pre-mRNA that then needs to be modified. And then RNA polymerase 1 transcribes the other 3 ribosomal RNAs. So the question I asked you on the study guide was this: how do the various types of RNA function in protein synthesis? And we've got mRNA, tRNA, and rRNA. Those are the 3 types that you need to know how they function in protein synthesis. And we'll give you that answer in just a moment after I give you the answer to the question I asked during our lecture break time. Okay, so all of these features are unique to eukaryotes. The Tata box encoding sequences, primers unique to RNA type. And so here's a molecular representation of the central dogma of molecular biology. Here's the DNA with its particular coding gene. That gene is then read and written into an RNA version of that gene, specifically mRNA, and then that gets translated into protein. Okay, so here is a molecular representation of the central dogma of molecular biology. But again, we know now that if you are not making mRNA, you're never going to go to here. Okay, and the central dogma does not explain even close to the full amount of what's going on. Okay, again, less than 2% of your DNA conforms to the central dogma. All right, here's your answer to your lecture break question. With another question. How does transcript modification add complexity? So a very simple way to answer the question I gave you over the lecture break is cutting out those introns adds complexity to this process. And you're like, cool, like that's a functional role of it. It's just to add complexity. But how does it do that? Glad you asked. And here's your real answer. A single pre-mRNA can be spliced in different ways to make different mature RNAs, to make different mature mRNAs. So what might be an intron for one type of mRNA might be an exon for a different one, and they were both made from the same transcript. A good example of this is the proteins that are written to make antibodies. Your body basically needs to be able to make an antibody against any foreign material that it should encounter. Okay? Okay? Which leads to, if your immune system isn't matured properly, to overreacting to things that it shouldn't, like pollen, or what are some pet dander, right? Or peanuts, okay? Some protein from the peanuts. Or the proteins from egg whites, even worse than being allergic to peanuts. Because peanuts you can avoid somewhat easily, right? Eggs are really difficult to avoid. Anyways, sorry. And so the proteins that are used to build antibodies, they can be spliced in hundreds of different ways to make literally millions of different types of antibodies. So this increases what we call, or the size of what we call the proteome, which is all of the different proteins that a cell is capable of making. That's what the proteome is. So this increases the size of the proteome, again, the collection of all of the proteins a cell makes, without increasing the size of the genome. Because the same section of DNA transcribed into the same pre-mRNA can end up being translated into literally hundreds, if not thousands or millions of different versions of that protein. Instead of having to have hundreds or thousands or millions of different versions of that in the DNA, you have one. Well, I mean, technically you have two, because one on your maternal chromosome and one on your paternal, right? So you've got two versions of this that can produce literally millions of different types of protein based on how those introns get cut out. David and then Micah.
What's a proteome again?
Proteome is the complete set of all proteins an organism makes. Yep. And so there's an idea. Oh, here, I'll tell you this. There's an issue. So if your view of origins holds to that the diversity of life is entirely the result of random processes, that there is no creator responsible for any of the diversity of life, you need to generate an enormous amount of diversity, right? Because you didn't start with any. You started with a single type of life, and then you've got to generate all of the diversity, right? You don't have this issue if you've got a creator that created some of this diversity because you're starting with some. Okay, but if you've got to account for all of it, you need a mechanism by which you can get really big differences with a very small change in the DNA, right? Because the whole drive of variation is what? What drives the variation? Mutations, right? And then natural selection selects certain mutations and generates not diversity. Diversity is generated by the mutations, but generates isolation so that you can actually get unique organisms that are no longer reproducing together. Okay, but if you're going to have a single mutation in the DNA that's going to result in something that natural selection can use to create isolation, it's got to have a big change. And the idea here is, well, if you can have a single mutation and then have this spliced in different ways and have it result in dozens or hundreds of different protein products that have never existed before from a single mutation, here's a nice way in which this might have happened. Micah, did I ever answer your question?
No.
I said David and then Mike, and then we never got. I'm so sorry.
Oh, yeah, but when intron gets cut out, then it splits in all those proteins, or
well, then it has to be translated. So we haven't talked about translation. The introns get cut up and get recycled. So once an intron gets cut out of that pre-mRNA, that little section of RNA gets cut up into the individual nucleotides and recycled for future rounds of transcription.
So I guess I'm still confused with why there's introns.
Because it allows you to write the same product, right? And so here, let me show you this. Here's a visual representation of it. So here's our pre-mRNA. Here's our pre-mRNA, okay? And it allows us to, by cutting this out, cutting out different pieces. So for instance here, we've got this pre-mRNA, and it cuts out everything except for 1, 2, 4, 5, 6, 7, 8, 9, and 12. So it cuts out, what would that be, 3, and it cuts out 10 and 11. You see that? They got cut out. And so when this gets translated, it makes a completely different type of protein.
Oh, I see.
Whereas when this cuts out 2, it cuts out 12. Is that it? Just 2 and 12? So this one cut out 2 and 12, and then when it gets translated, it produces a different type of protein. But you had the same gene, the same DNA code, resulting in 2 completely different protein types. And so that's the value of having sections that can be cut out. Because again, if they get cut out, they never get translated. And the analogy of like a letter, it's such a wonderful analogy for this, right? You write somebody a letter in a language that they don't know and they have to translate, but then there are different words you cut out before they translate. And so you could cut out every 5th word, translate it, right? Or you can cut out every 3rd word and translate it, and your translations are going to be completely different, but it all came from the same letter. Yeah, Chris.
So both proteins that each one of those code for, either like, you know, they cut out some of the proteins and then make one, or some of the
yes, some of the introns, and then it get, yeah. I mean, once you cut out the introns, then you're like, okay, now this is mature mRNA. It can leave the nucleus and get translated.
And so either one it gets translated into, the body actually uses both?
Yes, in different tissues.
Okay.
So it uses this version of this alpha tropomyosin in smooth muscle, and it uses this version of alpha tropomyosin in skeletal muscle.
And can there be more than 2?
Oh yeah, and that's like going back to the proteins that make up antibodies. I mean, you could literally generate millions of different types of them in the same cell, because there's a single cell type that makes antibodies. They're called B cells, and they can make, the same B cell can make millions of different types of antibodies. Yeah.
How is there 2 like variations when like the exons are being shared? Like the smooth muscle has 1, and then the skeletal muscle also has the number 1 exon.
Right, but they don't have all of the same. So the very beginning of these proteins, when this gets translated, the very beginning of these proteins are going to look exactly the same. But by the time you get to here, this protein is now going to look different than this one did.
Okay.
Because 2 follows 1 here, 3 follows 1 here.
Oh, okay.
So at the beginning of the proteins, they are going to look identical. But once you get to that second position, your protein's going to be different. Okay. And it's going to behave different, right? If you change the primary structure of a protein, it's going to change its secondary structure, right? Regional 3-dimensionality. And then it's going to change its tertiary structure, global 3-dimensionality. And everything about a protein functioning is how it's shaped. Okay? Make sense? Okay. Our next set of questions. Look at this. How does each RNA type function in translation? This is exactly the question that was on the study guide. And I told you last time on Monday, I said, this is a really good question. Maybe was it on Monday or was it last week on Friday? I don't remember, but I told you that this is a really good question on the study guide. I mean, you know, if I were you, I might anticipate having a question that looked very, very similar to this on the exam on Friday. And this, you're like, well, how's it going to show up? And this could show up as a matching question, right? Where you've got tRNA, rRNA, mRNA, and you've got to match it to its role in protein formation. Or it can show up just like this. And how does each RNA type function in translation? Wouldn't that be wonderful if it showed up just like this? After I've told you now, well, I told you one in previous class, and I've told you 4 times today. This is a good question. And you laugh, but even if I put this like this right on the exam, half of you will either skip it or answer it incorrectly. I'm sorry for smiling, it's not funny. How are tRNA and ribosomes formed? What are the 3 stages of translation? And then finally, what is left to make protein? After the process of translation, we don't have protein yet. You have a polypeptide, but you don't have a protein yet. Because something has to happen. Actually, several somethings have to happen. Okay, this first one. This is This is a really important question. How does each RNA type function in translation? Now, to transition from DNA to RNA is very simple. The language doesn't change. The letters you use change, but the language doesn't change. Right? The nucleotides are a little bit different. They're ribonucleotides instead of deoxyribonucleotides. There's no thymine, but there's uracil. But the language is very, very similar. The machine is similar, right? RNA polymerase, DNA polymerase, those machines are very similar. RNA to protein, it's not the same. It's very complicated. The language is completely different. So let me give you an analogy. It's not a great one, but it's the one that just came to my mind. So DNA to RNA would be like talking to somebody that speaks the Queen's English, right? You know what I mean by the Queen's English? Some of the words might be a little bit different, but the language is the same. Like, I don't understand what you mean by napkin, right? Napkin's a diaper in the Queen's English, I think. Is that right? Anyways, a nappy? Yeah. And so, like, the words are a little bit different, but the language is the same. Here, it's like trying to translate from English to, I don't know, what's the most different thing from English? Like Mandarin Chinese, okay? The language is completely different. The alphabet's different, right? Everything is different. Is that okay? I told you it wasn't that great of an analogy. It feels weird in here. So we have what's called the genetic code, and this gives us an ability to translate from RNA into protein. Okay, the genetic code. And here's the genetic code. Every 3 nucleotides, we call this a codon, codes for an amino acid. So every 3 nucleotides, a codon codes for 1 amino acid. And you're like, that's so cool. So let's play a little game. It's not really a game. It's more of a visual. So we've got a codon here that consists of 3 nucleotides. In this first position, how many options do we have for the nucleotide here? 4, right? It could be guanine, it could be adenine, it could be cytosine, or it can be uracil. Can't be thymine because there's no thymine in RNA. So we have 4 options here. What about at the second position? 4 options. What about at this third position? 4 options. And so if we want to figure out the total number of codons that you can produce, we'll do 4 times 4 times 4, which gives us what? 64. 4 to the 3rd power. So there are 64 different words in our RNA language. And from these 64 words, you can make the 25,000 proteins that exist in your cells, plus more. And you're like, that's so cool. How does that work? Let's see. All right, so now let's start talking about these RNAs, because you're like, you still haven't told us how these RNAs function in protein synthesis. Well, tRNA brings the amino acid. So the amino acid is physically attached to tRNA, and tRNA comes and it has an anticodon for the codon. So let's say, hypothetically, our codon is this, AUG. Okay? So this is a hypothetical. It could be anything, but this is AUG. A tRNA would come in, and the tRNA has an anticodon, and if the anticodon matches this, it will deposit its amino acid. And so the matching anticodon would be UAC, right? So the tRNA, it's got an anticodon, and if it's complementary here, it's going to give that amino acid to this growing protein chain, polypeptide. I used AUG because that happens to be the start codon, but we'll talk about that next week. Not on Friday, because on Friday we're not going to talk much. Although when I proctor exams, which I will on Friday, I get bored, and I'll talk more than I should. Just a warning. Ribosomal RNA, so tRNA has the anticodon, it brings the amino acid. Ribosomal RNA makes up part of the ribosomes, and it provides the catalysis. It provides the energy necessary. ATP provides the energy necessary, but it hydrolyzes ATP to give you the energy necessary to attach these amino acids together. And what type of bond links amino acids together? We call it a peptide bond. And what type of reaction is required to make a peptide bond? It's the same reaction type that we use to make all of our macromolecules. And it's another question on the study guide. Condensation dehydration reaction. Okay? So that reaction is carried out by ribosomal RNA. That dehydration reaction to form those peptide bonds, to string those amino acids together. tRNA brings the amino acid. Ribosomal RNA takes that amino acid and attaches it to the growing polypeptide. And you're like, well, what about mRNA? What role does mRNA play? It's bringing the codons. So here's mRNA, and it's bringing those codons. Okay, you see that? It's bringing the message that needs to be translated. tRNA, with its anticodons, is bringing the amino acids, and ribosomal RNA in these ribosomes are catalyzing those condensation / dehydration reactions. So let's just say this question appeared word for word on your exam on Friday, hypothetically. If you were to answer that question by drawing this picture, I would be satisfied. So hypothetically, that question comes, you draw this picture, I would look at that, I would smile, and I would give it a 10 out of 10. And you're like, well, I don't really want to draw a picture. I'll just write it out. And then so I'll write that tRNA brings the amino acid, mRNA brings the message to be translated, and rRNA facilitates the condensation reaction. I'd smile for that too. You know, just hypothetically, if this question does appear word for word on Friday's exam. Okay? It's 12:40. We're out of time. We got exactly to where we needed to get to today. Well done. Have a wonderful day.